Hello everyone, let's get started. Today we're going to be talking about muscle cells and contractility. We're going to begin our discussion with contractility in skeletal muscle. So far as we've been talking about the way things move around inside a cell, we've discussed the components of the cytoskeleton and we've talked about different types of motor proteins, kinesins and dyneins. We've talked about how these motor proteins can walk along microtubule pathways along relatively long distances inside of a cell. Now we're going to switch gears and we're going to talk about a different kind of movement. It's a movement along a much shorter distance. It's a movement along not microtubules, but instead along actin that has been arranged into what's called a thin filament. Thin filaments are analogous to the microfilaments that we talked about in our discussion of the cytoskeleton. The type of motor protein that performs this type of short distance movement is called myosin. Myosin is another one of these ATP-dependent motor proteins. And like the kinesins that we looked at, there are many different kinds of myosins. In fact, there are 20 different classes of myosin protein. But they all share some features in common. Structurally, they look a lot like kinesins and dyneins. They have globular heads, they have heavy chains, they have light chains. They also provide directional movement. On this slide, you're looking at a table that you've seen before. I showed you this table when we were talking about kinesins, so that we could see that there were different families of kinesins in cells. Well, if you go down to the bottom of the table now, you can see different families of myosins as well. We're going to be focused in on the first type, which is myosin type 2. This is the type of myosin that you find in contractile cells. Type 2 myosin is one of the most heavily studied proteins that there is. We know an awful lot about what it looks like structurally and how it functions. It has two globular head structures associated with it, two heavy chains, and two what are called light chains. We've seen this type of language before associated with motor proteins. Over here on the right-hand side of the slide, you're looking at a drawing of a myosin type 2 protein. The globular heads are sticking off of one end of the protein. The double coiled helical chains are found leading off from those globular heads. And we can see some light chain regions in between those two structures. Myosin type 2 is found in skeletal muscle cells, smooth muscle cells, and also in some non-muscle cells. This is a motor protein that converts the energy that is stored in ATP into mechanical force, just like we've talked about with other motor proteins. And what myosin is going to do, rather than traveling again down some long distance, along these actin thin filaments, what myosin is going to do is move the actin thin filaments. It's going to slide them across short distances in order to achieve contraction of the muscle cell. When you compare myosin type 2 and kinesin, they share some things in common. Again, they are structurally very similar. They both have these globular heads that do the actual movement for the motor protein. And again, they both use ATP as a source of energy. They hydrolyze the ATP. But myosin and kinesin have some important differences as well. Remember, kinesin operates alone. Each kinesin molecule carries its own individual cargo. Now, when there's a lot of cargo to be moved in a cell, you can see groups of kinesins traveling down microtubules, but each individual kinesin molecule is working by itself. Myosin, myosin type 2 specifically, works very differently. Myosin works in groups, in what we call arrays. Myosin is going to bind to these thin filaments, these structures that look a lot like microfilaments, and it's going to slide them. As the myosin moves, it's going to force the thin filaments to move. Now, this is a relatively short distance movement. It's about 12 to 15 nanometers each time the myosin moves. It's going to move in these short bursts referred to as power strokes. So myosin is the motor protein that's going to allow muscle cells to have this characteristic that we refer to as contractility. Let's take a few minutes now and look at muscle cells in terms of their anatomy and how they are arranged into what we think of as muscles. Muscle cells, first of all, are not referred to as cells. They have a very unusual shape. It's elongated, and so we refer to them as fibers. So all a muscle fiber is a cell. It's an individual cell that happens to have that elongated linear shape. Now, when you take a single muscle fiber and you look inside of it, most of the cytoplasm is full of these structures referred to as myofibrils. Myofibrils are very abundant inside of each muscle fiber, and they, again, take up most of the room inside that muscle fiber or that muscle cell. Now, if you take one of these myofibrils out of a muscle fiber and you examine it, it's actually composed of bundles of filaments that are both thick and thin. Now, in skeletal muscle, specifically what we're talking about first, these thick and thin filaments that make up the myofibril are arranged in very particular patterns, overlapping each other in areas, and they create a very specific, very orderly pattern of striping down the length of the myofibril. Now, the technical word for this striping pattern is striation. So that's why we say that skeletal muscle fibers are striated. They are literally striped when you look at them under the microscope. And that striping pattern, those striations, come from the overlapping pattern of the thick and thin filaments inside the cell. So on this slide, if you start up at the top, you're looking at a drawing of a person's arm and some of the muscles that are found in the upper arm, including that muscle that's called the biceps brachii. Now, we think of this large, grossly visible structure as a muscle, but in reality, this is a very complicated, elaborate, organized grouping of muscle fibers, muscle cells, in other words. If you were to look at this muscle in terms of these organizational levels, what you would see under a microscope is groupings of individual muscle fibers, something like this. So what you're looking at here, what looks like a group of sticks or maybe cigars. Somebody said they look like little Italian cookies. all lined up together on a plate. These are groupings of muscle fibers. It's a bundle of muscle fibers. So each one of these linear structures is a muscle fiber or a muscle cell. And one of the things that you notice about them is, number one, they're very strangely shaped. They look like tubes. They don't look like the kinds of spherical animal cells that we're used to seeing. Number two, they have stripes on them, and that's unusual. That's the striation pattern that we talked about a minute ago. Now, if I were to take one of these muscle fibers, one of these tubes, and blow it up, that's what I'm looking at here. So this is one cell, one muscle fiber. You can see the striations clearly from the outside. You can see a nucleus right here, sort of squished up against the membrane. And you can see that the membrane has started to be cut away here so that we can look into the cytoplasm. And basically, the cytoplasm in here is full of more linear structures called myofibrils. So each cell is full of myofibrils. And then when we group a bunch of cells together, many cells together, we get what we think of as a muscle. Now, if I took this myofibril and I examined it closely, it would look something like this. Along the length of the myofibril, I would see that very regular striping pattern. And underneath the microscope, I would see a combination of thick and thin filaments arranged in an overlapping, repeating pattern. This is what we call a sarcomere. So the sarcomere is made out of thick and thin filaments that overlap each other at intervals. Many sarcomeres together make up a myofibril. Many myofibrils together make up a good part of a muscle cell or a muscle fiber. Many muscle fibers together make up what we think of as a muscle. Skeletal muscle fibers are unique not only because of the way they're shaped, not only because of their striations, but also because of some of the other anatomical features they have, structural and functional features that they have. One of the most interesting features is that these are multinucleated cells. So far as we've been talking about eukaryotic cells, we've said that each individual cell has a nucleus along with other organelles, and the nucleus is where the DNA is housed. Well, a skeletal muscle fiber has several nuclei, and the reason that it has several nuclei relates to how muscle fibers are put together in a developing fetus. Several individual immature cells will come together and merge to create one muscle fiber. So when that muscle fiber matures, as the fetus continues to develop, it's actually composed of several individual cells. So these are multinucleated cells. That's something unique among the cells we've talked about so far. The plasma membrane that surrounds a muscle fiber is highly specialized, so it gets a name. We call it the sarcolemma. Anytime you see this term sarco, that relates to muscle. Inside the muscle fiber, there is a specialized organelle called the sarcoplasmic reticulum. So this is another reticulum similar to the endoplasmic reticulum that we talked about. Remember, we said that term reticulum means net or mesh. So the sarcoplasmic reticulum is also going to have that net-like structure. And it's going to surround all of those myofibrils that we talked about. It's going to travel around the outside of all the myofibrils, and it's going to dive in and around all those myofibrils. The job of the sarcoplasmic reticulum is to store and release calcium ion. And it's the calcium ion that's going to help regulate contraction of the muscle fiber. Now, we said the cytoplasm is packed full of myofibrils. That's also an unusual feature for a cell to have. These myofibrils, remember, are composed of a combination of thick and thin filaments. And those filaments are arranged into sarcomeres. The sarcomere is what we consider the fundamental unit of muscle contraction. So contraction begins with the sarcomere. And many, many, many individual sarcomeres are found in each muscle fiber. And of course, many, many, many muscle fibers are found in each muscle. So the unit of contraction inside that big biceps brachii muscle, the unit of contraction is the sarcomere. One other feature of muscle fibers to note is that they contain a protein called myoglobin. Myoglobin is a protein that can bind gases like oxygen and hold on to those gases. Now, like the name suggests, myoglobin is related to the protein that we've talked about before called hemoglobin. These two proteins share structure in common. They both contain heme units, and it's the heme that can bind to gases like oxygen. The difference between myoglobin and hemoglobin is that hemoglobin is found in the blood. Hemoglobin is found in red blood cells. Myoglobin is not found in the blood. Myoglobin is only found inside muscle fibers. The myo term here also means muscle, just like sarco does. So it would be abnormal to find myoglobin in the blood. The only time you would find it in the blood is if skeletal muscle fibers had died off or broken down and released the myoglobin. Now, having myoglobin inside of the muscle fiber is an advantage because it can bind and store oxygen. So under conditions where not enough oxygen is coming into a muscle fiber to meet the needs of that muscle fiber, the myoglobin can release its oxygen stores and bridge at least a temporary gap in fresh oxygen supply from the blood. On this slide, down at the bottom, we're looking at some skeletal muscle fibers underneath a light microscope. Up at the top, we're just looking at a drawing of a skeletal muscle fiber. Part of the sarcolemma has been removed, so we can look inside the cell. Now, if you start down at the bottom, one of the things you notice, and I haven't told you what magnification we're at, but one of the things you notice is that these cells are big. We normally expect to see many, many circular or spherical cells underneath the lens of a light microscope. We're looking at multiple cells here, but they're big. We can't even see the entire cell. We're only seeing a portion of it. So there's a cell here, and here, and here, and here. These are linear, elongated cells, and they're large. The next thing you notice is that there are many nuclei present in each one of these cells. That's also quite unusual. Now, it's a little hard to see, but if I enlarge the image, I think you can see the striations in these cells. That's visible even underneath the light microscope. Remember, each muscle fiber is striated because it's full of myofibrils that are striated. And the reason the myofibrils are striated is because they are full of filaments that are arranged into sarcomeres. It's the sarcomere, ultimately, that's responsible for this striping pattern that we can see in these cells. Now, when you go up to the top, you're reminded that the reason that the cell is striated is because the myofibrils are striated. And you can just see in this drawing individual sarcomeres. You can see a bright red line right here, for example, in this myofibril. And then if you move up, you can see another one right here. The distance from this bright red line to this bright red line, that's one sarcomere. So the sarcomeres are lined up end to end in each one of these myofibrils. And there are many myofibrils inside this muscle fiber. You can't see the nuclei in this drawing. You can't see the other organelles in this drawing. You can't see the sarcoplasmic reticulum in here. But it's a good reminder that most of the space inside of muscle fibers is just taken up by these myofibrils. So each striation in a muscle fiber is due to the presence of all those myofibrils inside. And the reason the myofibrils are striated is because they contain thick and thin filaments arranged into sarcomeres. It's the sarcomere that is the unit of contraction inside of muscle tissue. So the thick filaments and the thin filaments are arranged in sarcomeres so that they overlap each other. Let's take a look now at what these thick and thin filaments really look like. The thick filament is actually made out of myosin type 2 proteins. Now, I'm going to refer to myosin type 2 from now on as simply myosin. One thick filament is made out of lots of individual myosin molecules joined together. The thin filaments are made out of actin, as I said a few minutes ago. And there will be some associated proteins that work along with that thin filament in the sarcomere. When you take a myofibril out of a muscle fiber, like this one, and you cut it in a transverse plane so that you're looking at a cross section, you would see something like this. You would see the face of that cross section, and you would see all of the thick and thin filaments inside. What you're looking at down at the bottom here is that face, that cross section through a myofibril. And this is an actual image taken with an electron microscope. The reason I put this up here is to show you that while we often think about sarcomeres in 2 dimensions, we look at pictures of sarcomeres drawn in 2 dimensions. In reality, these filaments are arranged in 3 dimensions. The thick is surrounded by thin filaments in 360 degrees. And it creates this really amazingly orderly pattern that is the myofibril. So in this hexagonal organization, everywhere you see the open black circle, that's a thick filament. And all of the smaller, grayer structures around each thick filament are thin filaments. So for each thick filament, there are 6 thin filaments surrounding it in a hexagonal pattern. And the entirety of the myofibril is made out of these orderly arrangements of sarcomeres. Now, the scientists who first discovered the sarcomere and the patterning of thick and thin filaments noticed that underneath the microscope, the sarcomere had very particular patterns of banding within it. And they described that banding using some very simple lettered language. They talked about an A band and an I band and an H zone and so on inside of a sarcomere. And they based those bands and other anatomical structures on what they looked like underneath the microscope. In other words, they didn't know what was in an A band, but they could see the A band under the microscope. We now know what an A band is. And so while we still use these terms, we can now add in what each one of these structures is actually made out of. For example, the A band in a sarcomere is the dark band, and I'll show you a picture of this in just a minute. And what that dark band is made out of is thick and thin filaments overlapping each other. Again, I'll show you this in just a moment. There's also an I band in a sarcomere. That is colored quite light, and it's created by the places in the sarcomere where there are only thin filaments overlapping each other. Now within the bands, there's something called a Z line. This is a dark line in the center of the I band, and this is a structure where all of the thin filaments are anchored. There's also an M line in the center of the H zone. I know, a lot of new terms. And it's the M line where the thick filaments are anchored. So the thin filaments are anchored to a Z line. The thick filaments are anchored to an M line. And the last location is the H zone. The H zone is a light region in the center of that dark A band. And the H zone is made from thick filaments overlapping each other. But these thick filaments have no globular heads in this particular region. Let's take a look at these anatomical locations inside of a sarcomere. At the bottom of this slide, you're looking at a drawing of a sarcomere, again in 2 dimensions. And up at the top, you're looking at a sarcomere underneath an electron microscope. Remember, when we looked at the drawing of the myofibril, we said that sarcomeres run from one bright red line to the next bright red line. Well, in the drawing down below here, those bright red lines would be located here and here. So from here to here is one sarcomere. Now, these lines get a name. These are the Z lines. And we said that Z lines are the place where the thin filaments are anchored. So these blue lines here that are horizontal, those are thin filaments. And you can see that they're all anchored in to this vertical Z line. Now, this black line right here, that's the M line. And that is the place where these brown structures are anchored. And these are the thick filaments. Remember, the thick filaments contain myosin, and myosin has the globular heads. That's what you're seeing jutting off of that brown linear structure. Now go up to the top for a minute. This is the Z line here. Here's another Z line here. So from here to here, that's one sarcomere. Notice these dark bands right here. From here to here, a very dark region. That's the A band. Now within the A band, there are thick filaments and thin filaments overlapping each other. That's what gives it the dark color. And if you follow down to the drawing, you can see this region. It stretches from here all the way over to here. There are brown and blue lines overlapping each other, thick and thin filaments overlapping each other. The very light region in the center of the A band that has the black line through it, this is the H zone, and the black line is the M line. The H zone is an area where only thick filaments are found, and it's an area where those thick filaments have no globular heads jutting off. The M line, again, is where those thick filaments are anchored. And finally, we have the I bands. You can see that an I band actually stretches from one sarcomere past the Z line into the beginning of the next sarcomere. The I band has a very light color because it's a region where only thin filaments are found. Only thin filaments are overlapping each other. And of course, the dark line in the center, that's the Z line, where those thin filaments are anchored.